Okay. So here we have 3 water molecules, and each of these 3 molecules is made up of an oxygen atom and 2 hydrogen atoms. Okay? Oxygen is what we refer to as a very polar element, and that means that it likes electrons. It has a high electronegativity, which means that it draws electrons towards its nucleus, and it takes those electrons more often than not from hydrogen. At any given time on the oxygen atom, there are more electrons orbiting than around the hydrogen atom. So this is what causes the V shape of the water molecule. You can see here each of our 3 water molecules have this distinguished V shape. This is what it means for the molecule to be polar. Now there are 3 elements that are mentioned in the lab manual introduction that usually indicate that a molecule will be polar. And those 3 elements are oxygen, nitrogen, and phosphorus. And the reason being is that they are all electron hogs, very electronegative. So here we have these 3 oxygens causing this V-shaped bend in the water molecules, and water is a polar molecule. It is an asymmetric molecule. That means that there is not a symmetry to the molecule. Now we're going to contrast that to the next molecule, and the next molecule that I'm going to show you is one that is comprised of carbon and hydrogen. This is called methane. This is methane gas. And this is comprised of a carbon atom and 4 hydrogens. So 4 hydrogens surrounding that carbon atom. They're all equally spaced around that carbon atom. Okay. Notice that there are no oxygens, nitrogens, or phosphorus elements. That indicates that this molecule is nonpolar. Nonpolar because it's symmetrical, nonpolar because it's lacking any of those 3 elements that indicate that polarity. Okay. This is methane in its natural form, methane gas. This is the smallest hydrocarbon. Now, as we build up and we get a little bit more complicated, we're going to see, we're going to connect the 2 carbons and put them together to make ethane gas. Now, ethane gas is made up of 2 carbons and 6 hydrogens. Okay? Again, this is a molecule that is symmetrical. It is nonpolar. Notice I can rotate these 2 carbon atoms around the axes, so there is some flexibility there. And this molecule here is also related to the next molecule I'm going to show you. I'm going to put them side by side. This is called ethene. And ethene has 2 carbons, just like ethane. But notice what's different is that these 2 carbons are held together by a double covalent bond. Whoops. So that double covalent bond actually then decreases the number of spaces for hydrogen to bond to the carbons. So instead of having 6 carbons like you have in ethane gas, now you're down to 4 carbons, okay, with that double bond. I wanted to show you this does not have that flexibility. I cannot rotate these carbon atoms around each other because that double bond is actually a much stronger bond than a single covalent bond. Okay. And finally, we have 2 carbons here that are held together by a triple covalent bond. So 3 bonds holding together those carbons. I cannot rotate those carbons, atoms, around each other, and there's only 2 hydrogens connected to this molecule. This is acetylene. And notice that as I increase the number of bonds between the carbon atoms, I'm decreasing the number of hydrogens that are attached to those carbons. Okay. All of these molecules here are nonpolar. Nonpolar. We don't have any oxygens, we don't have nitrogens, we don't have phosphorus. And nonpolar also means that they're not dissolvable or don't mix well with water. Okay, so these are relatively small molecules. The next molecule we're going to take a look at is a little bit larger. And this is a chain of carbons attached to hydrogens. So it's still a hydrocarbon chain. Notice how different this shape of the molecule looks from the shape from the molecule that's drawn in the lab manual. In the lab manual, you'll have this very linear structure. This almost looks like a zigzag or a zipper structure. And this is the space that carbons take up in real life. So they're not a chain that's straight and linear, but rather a zigzag or a zipper shape. And that's just to be able to come in close proximity with all of those carbons and those hydrogens. All right, so here we have a chain of 6 carbons and 14 hydrogens. Those hydrogen, this is a long hydrocarbon chain. These are our basis for all of our biological molecules. A long chain of carbons and hydrogens are what constitutes proteins and carbohydrates, nucleic acids, and lipids. This is the base for all of those 4 different types. Category that we're going to take a look at are proteins. And proteins are made up of the subunit amino acid. And in order to build an amino acid, we need 2 functional groups. The first functional group is an amino group. And an amino group is made up of a nitrogen and 2 hydrogens. As we discussed earlier and talked a little bit about and intimated about the polarity of a molecule, nitrogen is one of those elements that indicates that a molecule would be polar. And indeed, an amino group, nitrogen and 2 hydrogens, is very polar. The second functional group we need to build an amino acid is called a carboxyl group. And that carboxyl group is made of a carbon, 2 oxygens, and a hydrogen. Notice that the oxygen and carbon have a double bond. This is also a very polar functional group. Okay, so these 2 functional groups are needed to make up an amino acid. And an amino acid is made up of a central carbon atom, and it's got 4 covalent bonds coming off of it. What we're going to do is attach this amino group to that central carbon atom and attach that carboxyl group to the central carbon atom. And this is what an amino acid looks like. I think it looks like a little poodle dog. Okay. So this is the backbone for building all proteins. All proteins are comprised of amino acids. Put them all together, and then you get a long chain of what we call polypeptides, many peptides. So the next step we want to show you is how you put these amino acids together. So I'm going to do my poodles on parade here. I have 2 amino acids. And those 2 amino acids, you notice they look kind of similar to each other as far as the R group, the amino group, the carboxyl group, and then the free hydrogen. The way that these 2 fit together is through a reaction called the dehydration synthesis reaction. This reaction involves the removal of a water molecule so that these 2 functional groups, the amino group and the carboxyl group, can come together. So I want to demonstrate this. You'll notice in the lab manual that these groups are highlighted, and they're highlighted to show you how these 2 molecules can come together. So this first amino acid here, what's going to happen is this OH group comes off, and on the second amino acid, the hydrogen atom of the nitrogen group comes off as well. And then this is where they will interface, where those 2 groups of atoms come apart. We're going to join together our 2 amino acids, and this is known as a dipeptide. Di meaning 2, 2 referring to the 2 amino acid groups. And what happens with that hydrogen and the oxygen and hydrogen group? They form water. Hence the name dehydration, removal of a water molecule from between these 2 amino acids. Now, this is a very small molecule, relatively speaking. Proteins are made up of hundreds of thousands of amino acids repeated all in a row. Every time your body goes to build a protein, it does this step-by-step process to build that protein. Okay? So these 2 molecules here, you'll notice, one is much larger than the other. They ask you to identify what this large molecule is. This is your dipeptide. And then this smaller molecule, obviously, you probably recognize from the very first exercise, this is our water molecule. Okay. Notice the backbone of our dipeptide has a repeated sequence here. We have 2 carbons and a nitrogen, 2 carbons and a nitrogen. Again, this is something that's common in any and all living organisms. Same backbone, same structure of all proteins. The next category we're going to take a look at are carbohydrates. And carbohydrates are long chains of carbons and hydrogens. Here we have what we call a linear glucose molecule. Linear because it's in a line somewhat. Doesn't look exactly like the lab manual. Again, you'll notice that zipper structure. We have 6 carbons in black. We have 6 oxygens in red. And then we have 12 hydrogens in yellow. Okay. Notice the carboxyl group at the end of this linear glucose molecule. And all of these hydroxyl groups, the hydrogens and oxygens, these indicate that the molecule is extremely polar. Again, we can see all those oxygens indicating the polarity of the molecule. Readily dissolvable in water. And if you think about some of the more familiar carbohydrates, such as glucose or sucrose or lactose, they're very much water soluble. This is why. Their chemistry and the chemical characteristics dictates that they are polar, they like water, they're water loving. Okay? Now, glucose, normally if it's floating in your bloodstream, doesn't look like this linear molecule. It's actually in a ring structure. And that ring structure is the next structure I want to show you. This is what we refer to as a cyclical carbon chain, just because it's attached in a shape. This happens to be like a hexagon shape, a 6-sided shape. Again, if I count up the number of oxygens, carbons, hydrogens, it's exactly the same as my linear molecule. We have 6 carbons, okay? But again, notice they're in a ring shape. We have 6 in red oxygens, and then 12 hydrogens in yellow. I'm going to hold this up just so you can see that central ring structure is hexagonal. This is called a hexose, or 6-sided carbohydrate. And this again is very polar. This is a molecule that likes water, likes to interact with water, readily dissolvable in water. Another molecule of a carbohydrate molecule that is a small carbohydrate is fructose. And fructose is found in, for example, your fruits like bananas or apples or oranges that makes it sweet taste. Fructose is a great form of energy for us. You eat a piece of fruit, you get a little bit of a sugar rush, a sugar bump. That's the fructose in that food. This is slightly different than our glucose molecule. Notice the shape of it. And again, I'm going to hold up the ring structure so you can see the pentagon in the middle. Whoop. That's a 5-sided sugar. We call it a pentose. So a nice 5-sided shape. The way that this is put together is slightly different than the glucose molecule because of that shape difference, but there's still 6 carbons, 6 oxygens, and 12 hydrogens. Okay, these are what we call isomers of each other. They have the same molecular formula, but they're slightly different in their arrangement. We're going to put together, next, we're going to put together the glucose and the fructose molecules. We're going to form what's called a disaccharide. Disaccharide means 2 sugars. And again, this is how plants, for example, will put together a glucose molecule and a fructose molecule to make sucrose. So for like sugarcane, for example, this is the way the plant would manufacture it. It's going to put together these 2 molecules the same way that we put together the 2 amino acids, which means that we're going to take a functional group off of one molecule and a functional group off the second molecule to combine them. Okay? Very similar to the amino acid forming into a dipeptide, we're going to take a hydroxyl group off of our glucose molecule. Take that OH group off here, and we're going to take off the hydrogen of the fructose molecule. It's stuck. There we go. And we're going to join them together without destroying the molecules, we hope. So they're bridged together here at this oxygen. This is where we're going to connect those two sugars or saccharides. So now, again, we have the leftover molecule that comes out, our water molecule, dehydration synthesis reaction. And now we have a larger structure, and this larger structure we refer to as sucrose. All right, our next category of molecules are the nucleic acids. And these nucleic acids, examples of nucleic acids would include DNA and RNA. DNA and RNA are both made up from nucleotides. And nucleotides are comprised of sugar molecules called ribose and deoxyribose. So deoxyribose is found in DNA and ribose is found in RNA. Both of these are carbohydrates, which means that they are long chains of carbon with OH groups attached, those hydroxyl groups. Very much a polar molecule, again, as you can tell by all those oxygens. This molecule is a molecule of ribose, and ribose has 5 carbons in black. It has 10 hydrogens in yellow, and it has 5 oxygens in red. So 1, 2, 3, 4, 5. So ribose is a little bit different than deoxyribose. You can see the highlight in the lab manual, just slightly different. Deoxy means without an oxygen. So what happens if you compare the ribose, which I have here, and deoxyribose, show you, this OH group comes off, and it's just replaced with a hydrogen. Okay? So this molecule here is deoxyribose. Slightly different because it has 1 less oxygen. So it has 4 oxygens instead of 5. And that's why we call it deoxyribose, without an oxygen. Again, very polar molecule because of all these hydroxyl groups on that carbon backbone, readily dissolvable in water. Okay. Our final category are the lipids. Okay, the last category are the lipids. And lipids are also comprised of long chains of hydrocarbons. What's unique about this category is that lipids tend to be hydrophobic. And hydrophobic means that they're water-fearing. It also means that these molecules are non-polar. So what I have here is a long chain fatty acid. And fatty acid chains are made up of a long chain of hydrocarbon, and then they have a carboxyl group at the end. Carboxyl group is that functional group we talked about earlier with amino acids. It's a carbon and 2 oxygens and a hydrogen. What's interesting about this molecule is that just because this carboxyl group is on the end does not mean that it is polar. So it is one of those exceptions to the rules that we've been mentioning in the lab manual. This long chain fatty acid does not like water. Now, one of the things I wanted to point out to you is that this long chain fatty acid has a double covalent bond in between. That means it's got some flexibility. It's got a little bit like a wiggleness to it. These long chain fatty acids that have a double covalent bond tend to be liquid at room temperature. That means they don't form solids. Okay? That's different from a long chain fatty acid that has all single covalent bonds. This type of long chain fatty acid we refer to as an unsaturated fatty acid. Unsaturated because there are less hydrogens on this molecule than the molecule underneath it. So I want to show you how to make a saturated fatty acid chain. We're just going to swap out this double bond here between the 2 carbons. Going to replace that double bond with a single covalent bond. And now we're going to add back those hydrogens because we don't have any holes, free holes on those carbon atoms. And now we have this last molecule, which is referred to as a saturated fatty acid chain. This is also still a nonpolar molecule. It's referred to as saturated because all of those hydrogens that are single covalent bonded to each other have the maximum number of hydrogens attached. Therefore, we say it's saturated with hydrogen. These molecules do tend to be solid at room temperature. So think about molecules that come from animal fats, for example, butter, lard. If you fry up bacon in a frying pan and you let that grease cool down, it'll congeal to form a solid. Okay, so this is the difference between saturated versus unsaturated.